Okay, let's first thank you for giving me an opportunity to share my research work and also discuss with our grad student as here and also our ajahn as here. So maybe it's not the lecture, but it's just like share the experience of my work during this period. So at introduction, you already know that I will talk about the extracellular vesicle or EV. So in the aspects of the Naegleria fowleri infection which is a free living amoeba, and I would like to see do they affect on the innate immune response or not. So first, I would like to give some little background about the free-living amoeba. As you may already know that free-living amoeba is a free-living organism that exists in the nature, such as water, soil, air, and some free-living amoeba are potentially pathogenic to humans and animals, but the infection are not commonly reported. However, there are 4 free-living amoeba that can cause disease in human and also are capable of the producing of CNS or central nervous system infection. For Acanthamoeba and Ballontia mandilaris can cause GAE or granulomatous amoebic encephalitis, which is slowly infections of CNS, and these are opportunistic pathogens. So Acanthamoeba can cause frequently in infections in the immunocompromised patients, and for the Ballontia cause frequently infections in immunocompetent children. And for the Naegleria fowleri, is caused primary amoebic meningoencephalitis or PAM, which is a rapidly progressive of the CNS infections. And this one, I will focus on these infections. And for the last one is sapropenia. Sapropenia is really like case in the CNS infection and is not lethal it's not lethal. So let me talk about Naegleria fowleri a little bit. There are 4 stages. There are 3 stages. First is cyst, tophozoid, and fractures. So how we get infection by the Naegleria fowleri? The patient come with the history that have activity in water, such as swimming, diving, or water sport. These Naegleria fowleri can enter through the nose and penetrate through the nasal mucosa and migrate to the brain via the olfactory nerve that can cause a primary amoebic meningoencephalitis or PAM. And one is lead to the brains. These tophozoites can like replicate and destroy some nerve cell and also destroy arterial cell tightness nearby. So via the non-contact in contact or contact dependent mechanism that I will talk later in the pathogenesis of the disease. Since I mentioned that this Naegleria fowleri can cause PAM, that is normally is rapidly infections and resulting in death maybe within 5 or 7 days. So the pathogenesis of PAM is associated with the pathogenicity of Naegleria fowleri itself and also the intense inflammatory response from the host immune cell. So the first one, the pathogenicity of Naegleria fowleri, they are divided into 2 groups. First is a contact-dependent mechanism that the Naegleria fowleri have some adhesion that can attack to the nasal mucosa, and also they have some the proteins that help. For example, that is have the variant factor that is of Naegleria fowleri have like a food cup that can like pseudopodiacs, so it can phagocytosis. So this one is such as NFA1. And for contact independent mechanism is like the protein that is can secrete, such as performing protein, hydrolytic enzyme, such as acid hydrolase, phospholipase, neuraminidase. So finally, this is makes the disease progression. And for the innate immune system, since our body try to defend against the Naegleria fowleri, so the major of immune system parts that have the major role is complements, neutrophil, and macrophages. For neutrophil, they have like myeloperoxidase hydrogen peroxide halide system that can defend against these infections. And for the macrophage, one of macrophage, which is microglia cell, which is brain tissue macrophage, can these Naegleria fowleri can induce the productions of the pro-inflammatory cytokine TNF alpha, IL-1 beta, and IL-6. But these inflammatory factors contribute to the disease progression. However, even we have the innate immune system that try to defend against Naegleria fowleri, but Naegleria fowleri very have the experts in the pathogen that is can develop to evade our host immune system, but they have some resistance to the complements such as C5b to C9, and also resistant to the cytolytic molecules such as the pro-inflammatory cytokine. And did photo show the neutrophil that Naegleria fowleri attack to the neutrophil. And also this one, the right hand side, is show that during the macrophage phagocytosis engulf the Naegleria fowleri. So let me talk about extracellular vesicle or EV. The research of EV start from 1969, and the spotlight of EV in the cell signaling system start from the 1996, that they present the work in the B cell, that they found that B cell can secrete EV, and this EV contains some MHC class 2 molecule that we already know that it have the major role in the antigen presentation. So this EV can help to the antigen presentation to the T cell. So from this, there are so many studies in the cell signaling system. For example, the hematopoietic stem cells, they also found the adhesions and chemotaxis, the functions of the EV. And after the boom of the molecular biology, so EV also have one study that around 2006 that they found that EV contains some RNA, and this RNA can transfer to recipient cell. So from this is very spotlight later on that there are so many study intent of the not only cellular part, but it's also molecular part. For the classification of EV, there are three type of EV depend on their size and biogenesis of EV. First, exosome, which is a smallest EV, size around 30 to 100 nanometer. This exosome occur formed by inward budding from the endosomal membrane to become the early endosome, and then it's become to intraluminal vesicle within the multivesicular body or MVB, and then they're filled to the plasma membrane, and this intraluminal vesicle can secrete extracellular space. So this one is we call exosome. Sometimes from the guideline, they just call like small EV. And the second one is the middle size, or what we call microvesicle, or sometimes they call outer membrane vesicle or large EV. This one is directly outward budding from the plasma membrane. So this one, the side length is around 100, not over 1,000 nanometer. And for the last one is apoptotic body, the largest EV that occur during program cell days with apoptosis. And for the right-hand side, you can see that not only animal can secrete EV, but fungi, plants, and also parasites can secrete EV. And as we know, the role of EV is a mediator in the intercellular communication and cell signaling. So they contain some cargo that can have their role and transfer to recipient cell. So for exosome and microvesicle or microparticle have very similar the cargo, but the outside surface receptor are different. So the content inside is just such as lipid, DNA, RNA, regulator molecule, cytosolic, cytoskeleton protein, adhesion molecule. And for the apoptotic body, they contain the base organelles such as DNA, RNA fragment, decay protein. And for the EV in parasitic research, nowadays there are many studies in parasite that they found that this parasite can secrete EV, such as Plasmodium species, Leishmania, Trichomonas, Toxoplasma, and also Acanthamoeba. So they are divided into three groups of the main, the source of EV in the parasite. First one is extracellular parasite, because this parasite can secrete directly EV to and induce immune cell and other host cell. The second one is intracellular parasite, because this one the parasite infect to the host cell, so the host cell the infected host cell can secrete EV, and this EV can respond to host cell. And the last one is extracellular parasite. They have like parasite antigen that can stimulate our host cell or immune cell and let our host cells secrete EV. And recently, that's the study in the EV in free-living amoeba. They study in Acanthamoeba castellanii. They found that these Acanthamoeba can secrete EV, and they found that this EV contain protease that is major virulent factor of the infections and can damage a whole tissue damage and host cell death. So from this study, let's us to would like to study in the EV in Naegleria fowleri to see that this Naegleria fowleri can secrete EV or not. This is the first question. And the second one, the effect of this EV to the host innate immune cell, monocytes, macrophage, and neutrophil. So my objective are divided into three parts. First, to isolate and characterize the extracellular vesicle derived from Naegleria fowleri or NFEV. And then I study the effect of isolated EV on THP1 monocytic cell line, which is a cell line. I use this one as a model for monocytes and macrophage. And later on, I studied the effect of this EV on the primary monocyte and neutrophil by isolate these cells from the healthy blood donor and see that this EV also affect on the monocytes and neutrophil. So in this study, I focus on small EV or what we call exosome, and large EV what I call the microparticle or MP. Let's start with the first one to isolate and characterize NFEV. So to isolate the EV, first we need to culture Naegleria fowleri, and then we isolate MP and exosome by sequential centrifugation and characterization EV by the nanoparticle tracking analysis or NTA and transmission electron microscopy or TEM. For the cultivation of Naegleria fowleri, we culture Naegleria fowleri with nice Nelson mediums in arsenic culture at 37 degrees Celsius, and when it's least around 80 to 90% conferency, we replace this media with paste amoeba saline without supplements for 24 hours before we collect supernatant. And two, there are many isolation methods, but we choose the sequential centrifugation to isolate the EV. First, we collect the amoeba saline buffer that from cultivation of the Naegleria fowleri, and then we centrifuge at around 1500 G to remove remaining tophozoite and cell debris. And later on, we filter first step with 1.2 micron filter, and then we centrifuge as to 20,000 G to collect the pellet of NP, and washing one step at the same speed and same time. And after we centrifuge 20,000 G, the supernatant we filter with the 0.2 micron filter, and then we centrifuge with anti-centrifuge at 1,000 G, 100,000 genes, and then we wash again, and then we get the MP and exosome to do experiments. For the characterization of the EV, first we characterize the EV by the nanoparticle tracking analysis or NTA. We can measure the particle size and concentration of particle in the cells in the suspension. The particle we detect, this NTA can detect the particle by the Brownian motion of the particle, and when laser direct to the particle, the NTA have like a camera detector that can detect the signal. This is I just capture exactly, it's like the keep video, but I just capture the photo that during we do in NTA, we can see the particle like this is and then its movement from flow during the flow rate. So the left hand side is MP and the right hand side is exosome. And this graph shows the diameter, the size, and concentration of MP. For the MP, we found that the size estimate around 150, and for exosome, the size is around 130. So when we compare the size of our EV to the study of Acanthamoeba, it's quite similar size, around 150. And when we characterize the EV by TEM, we found the cup shape and with the lipid bilayer of membrane of MP and exosome. And after we get the isolated EV, then we treat this EV to the THP1 monocytes and macrophage. So first we culture THP1 cells for 48 hours, and then we let cell for 24 hours. And after that, we co-culture THP1 with the EV. And for the macrophage, we co-culture with the PMN as stimulator, and then it's differentiated to the macrophage. So after we co-culture, we see the EV uptake by confocal microscopy, phenotypic characteristic by flow cytometry, and then cytokine production of this cell by qRT-PCR and ELISA. Let's start with EV uptake. So before I go to see the effect of EV, I need to investigate that do these EV can get inside the macrophage or not. So I culture this EV in ex-vivo cell culture slides for 48 hours. Culture this cell, and then for the EV size, I label this EV with PKH26, and after that I incubate together for 2 hours. And after that, if we stain the cell F-actin with the fluoridine with the color of green, and we stain the nucleus at the DAPI with the color of blue. And then we see the confocal microscopy. And this figure show the top is show the PBS control that is without EV. The middle one is the label MP, and the bottom is EXO, that is label PKS 26. If they have like MP and EXO, so PKS 26 is the less color, and we can see the color inside the cell. And when we look at inside, we found that both MP, this one is MP, can uptake into the THP1 macrophage and also exosome too. So after we know that EV can uptake inside the macrophage, so then we see the phenotypic characteristic by flow cytometry. But before we go through the phenotypic characteristic, we see the apoptosis first, because there are previous studies that they found that Naegleria fowleri lysis can induce the microglia cell apoptosis. So we have the question that do this EV affect on apoptosis of this cell or not? So we co-culture this EV for 48 hours, and then we stain the cell with annexin-5 and propylium iodide to see the apoptosis, because when the cell early apoptotic cell, this cell flip the phosphatidylserine to the cell surface, so annexin-5 can bind to the phosphatidylserine. So it shows that this one, if annexin-5 positive, is mean early apoptotic cell. While the propylium iodide is if the cell late apoptotic and the cell membrane lost membrane integrity, so they have some pore that propylium iodide can get inside to the cell and bind to DNA inside. So if the late apoptosis is represent in the annexin-5 positive and propylium iodide positive in both double positive. And what we found that percent total apoptotic cell, percent early apoptotic cell, and percent late apoptotic cell, we found that this EV did not induce in monocyte and macrophage in the apoptosis. So this result is different from the previous study that found that Naegleria fowleri lysis can induce the apoptosis of the microglia cell, but it's might because of the concentration that we use is different, because that study is very high concentration of Naegleria fowleri, and this one we use only 20 microgram per ml. And after that, we see the phenotypic characteristic of cell surface marker on the THP1 monocyte and macrophage by co-culture this EV for 48 hours and 72 hours, and we look at this CD marker. CD80, 86, and HLA-DR is a marker for co-stimulatory molecule that represent in the antigen presentation of, and CD169 is represent in the phagocytosis and also the antigen presentations. And after we stand this marker, we determine by the flow cytometry. And the right hand side is show the gating strategy. First, we get singlet cells, and then we get only the live cell by we stand with the zombie aqua to differentiate between live cell and dead cell. And after that, we compare these four markers by their isotype control. And this is what we found in THP1 macrophage. We found that the exosome can induce the expressions in the expression of the CD80 and HLA-DR, and for the MP can induce the CD80 only at 48 hours. And for 72 hours, we found that only MP that can increase the expression of CD86 and CD169. So it seems that the kinetic response of macrophage to this EV subtype were different between MP and exosome, but MP induced more prolonged effect than to the target cell than the exosome. And this is the result of the expression on THP1 monocytes. We don't see the upregulate expression on the THP1 monocyte, but we see just some trend in the CD169 expression, but it's not statistically different. And after we see the phenotypic characteristic, and we know that it can alter the expression of macrophage. So later on, we see that this EV can induce the cytokine production, especially in pro-inflammatory cytokine or not. First, we look at the cytokine expression at mRNA level by qRT-PCR. So we look skin for the cytokine expression of TNF alpha, IL-1 alpha, IL-6, IL-10, CXCL10. And the way that we do is we correct the cell that's incubated with this EV, and then we extract RNA and reverse transcriptase RNA to cDNA, and we amplify this cytokine expression by specific primer. And what we found, only TNF alpha and ILA were upregulated in the inductions of the exosome. So exosome might be potent factor during the Naegleria fowleri infection. And later on, when we look at the protein level by measure the cytokine production by ELISA, we found that only A that's upregulated in the MP when we induce the MP. So MP might induce macrophage activation resulting in the interleukin 8 production, and the role of interleukin 8 is chemotactic factor of the neutrophil. So it might involved in the induced migration of the neutrophil and causing accumulation these cells to the brain. So the study of neutrophil needs to further study. And the first part and the second part are our studies has already published data, and the last part is to study the effect of this EV on primary monocyte and neutrophil. We're still ongoing in this part, and we can show some data of the neutrophil. For the neutrophil, first we would like to see the effect of apoptosis, because neutrophil is a cell that's very short lifespan. They have like only 6 to 12 hours, and during the circulation, maybe it's just only 4 hours in the circulation. So we just wonder that this EV can affect on apoptosis of this cell or not, because this cell already spontaneous apoptosis by itself. So we co-culture this EV with the neutrophil for 4 hours, and then we scan for the neutrophil marker CD66B and also the enzyme 5 and potassium iodide to see the apoptotic cell. And what we found when we analyzed by flow cytometry for the percentage of early apoptotic cell, when we treat with CPT is camptothecin, which is a pro-apoptotic agent that can induce the apoptosis of cell. So this one can induce the neutrophil apoptosis. And when we look at the MP condition, we found that MP can delay the apoptosis of neutrophil when we compare to the unstimulated cell. So this neutrophil is mean that this delay neutrophil is mean that neutrophil is still in the circulation. So it might be maybe good for our host immune system that can like protect, or maybe it can other side that is disease progression in if this cell can produce the cytokine. So after that, we see the effect of the expression on neutrophil. Since normally in the blood circulation, neutrophil is in the resting state of neutrophil, but one get activated or primed with some microbial product or cytokine or chemokine. So it's become activated neutrophil or primed neutrophil. And to see that this one is activated or not, when it's activated neutrophil, they will shed, this cell will shed the alpha-lectin. So it means that it decreases the expression of CD60L, CD2L. And for the function of neutrophil, that is can degranulation. So if it's upregulation of this marker CD66B and CD11B, it means that the cell is activated. And when we look at the LPH, which is a microbial product that can prime neutrophil, we found that it reduce the expression of CD62L and upregulation of CD66B and CD11B, which is the marker for degranulation. And when we look at the MP and exosome, we found the same pattern. So it's this EV can activate neutrophil. So from my study, I study the effect of EV from Naegleria fowleri, and I see the effect of the on the macrophage that it can alter the aggregation marker on the cell surface, and also it can induce pro-inflammatory cytokines such as the ILA. So this data suggests that it may potential role in activating immune response that might be involved in driving the PAM pathogenesis. But later we will study more in neutrophil and also the primary monocyte, that we would like to see that are there any different or the same result as we see in the THP1 model or not. So at the end, this research was funded by the researcher Gan by McGill University, and I would like to acknowledge Professor Govit Pathana Panyasak from Sira Center of Excellence for Microparticle and Exosome in Disease, and from the researcher team in Sira, and also the Assist Professor Lada Wankova Visesut from the Department of Parasitology, and the researcher team and Ajahn from the Parasitology at Faculty of Medicine, Sira Hospital. Thank you for your attention.